Thank you for joining us for today's webinar, Advancing Patient Monitoring, Diagnostics and Treatment Strategies for Transplant Precision Medicine. This is the second webinar in our three-part Lancet series on transplantation, which is organized in collaboration with the Lancet and a joint presentation supported by the American Society of Transplantation, the European Society of Organ Transplantation, and the Transplantation Society. I'm Jen Lee and a transplant nephrologist at Westmead Hospital and a clinician researcher based at the Westmead Institute for Medical Research. It's my pleasure today to chair the webinar and introduce our speakers. So firstly, I'm delighted to welcome back Dr. Peter Reese, who has led and organized this series. Dr. Reese is a transplant nephrologist and he's a professor of medicine, epidemiology, and medical ethics. And he's also serving as the director of the Vanderbilt Center for Transplant Science. His research focuses on expanding access to kidney transplantation, improving the care of living donors, assessing the impact of health policies on the vulnerable populations with kidney disease, and developing strategies to improve health behaviors. He is a former chair of the UNOS Ethics Committee and elected member of the American Society for Clinical Investigation, just to name a few of his accomplishments. Today, we're also joined by our expert speakers, Alex Loupy and Marta Sablik, who alongside Peter co-authored the second review in this series. So Professor Alex Loupy is Professor of Nephrology and Epidemiology at the Necker Hospital in Paris. He is the founder of the Paris Transplant Group, the director of the Paris Institute for Transplant and Organ Regeneration, PTOR. He also leads the research team at the French National Institute of Health and Medical Research, INSERM, in transplant epidemiology. He chairs the BANF Scientific Committee and serves as an expert to the FDA and has made countless editorial contributions to high-impact journals. His work in precision diagnostics and AI-based risk stratification to transplant immunology has shaped international practice and policy, both in allo and xenotransplantation. He has received major national and European grants, including the ERC Consolidated Grant in 2023 for his project AI Care. Marta Sablik is a current PhD student at PTOR and INSERM, and this comes after completing her master's degree in medicine, infection and immunity from Erasmus University in Rotterdam. Her research spans kidney and lung transplantation, combining epidemiology and molecular approaches to study microvascular inflammation and to improve diagnostic classification of allograft rejection. Now, before I hand over to Peter, just a quick reminder to the audience. Please post your questions in the Q&A box, not the chat box, so that we can see and address them during the discussion. And now I'll hand over to Peter.
Thank you very much. I'm thrilled to be here. Well, I'm joining you from a cabin up in the mountains, so I'm going to go quickly since my internet connection is rather frail. But it's really exciting to lead this clinical series sponsored by the Lancet. Solid organ transplantation is life-saving for patients with end-stage organ failure. And three of the biggest challenges to our field have been with us since the beginning: a shortage of donor organs, the toxicity of the treatment over the lifetime of the person who is an allograft recipient, and inequities in access to transplant care. And we know that we have a very big problem globally that only 10% of the global need for transplantation is met. Today's session is the second in three. Last week, we had a session led by Matt Paquetta and Kira Shaver, which I highly recommend, which covered the many solutions that are coming at us quickly to expand the organ pool, advancements in organ preservation, repair technologies, and xenotransplant. Today, we're going to talk about immunosuppression, diagnostics, and the way that these tools are becoming more precise, allowing us to monitor the health of the graft in real time using non-invasive and invasive medicines, all leading us towards the goal of personalized medicine. And we're so fortunate to have two amazing experts in this. I also want to call out Kieran Kush, who is not here today, but made really foundational contributions to this manuscript and this content. And next week, we'll talk about equity in transplantation, both within countries and across countries. And then I just want to emphasize that this series is a foundation of knowledge that's brought us to today, and built on that will be the Lancet ESOT Commission on Transplantation, which aims to provide a comprehensive roadmap for the future of transplant medicine with policy recommendations, strategies customized to different areas, and support for global collaboration. So anyway, I'm very excited to turn this over to Marta and Alex, and I'm really inspired by their vision for the future of transplant and how patients everywhere will benefit from technological advancements.
Well, thank you, Peter, for the kind words, and thank you for organizing this webinar, of course. So before we dive into the discussion, I would like to just briefly outline the objectives of today's webinar, which will help frame our conversation today. So first, we will look into the major challenges that surround lifelong immunosuppression in transplant recipients, including the trade-off between preventing rejection and managing long-term toxicities. We will also discuss recent advancements in post-transplant monitoring, specifically invasive and non-invasive biomarkers that may help to tailor immunosuppressive regimens to individual patient needs. But we will also touch upon key research priorities that are actually essential for validating and implementing these novel biomarkers into routine clinic. Because we all know that many biomarkers are being studied, but we only see a handful of biomarkers actually being integrated into routine clinical practice. And last but not least, we will also discuss the emerging role of artificial intelligence, in which it can help us to integrate complex immunological and clinical data to better support patient monitoring, but also decision-making. And to ensure we're all on the same page, I would like to start with just a brief overview of the current practices in immunosuppression and patient monitoring. And as you all know, transplant recipients must take lifelong immunosuppressive medication to actually prevent graft rejection. And while these treatments significantly reduce acute rejection rates, they do come at a cost, causing serious side effects like infection, cancer, cardiovascular disease, and also metabolic complications, which is also nicely illustrated in one of our figures from a paper. And the standard approach today is really a one-size-fits-all. Patients receive similar treatments without really taking always the individual immune risk into account. And despite decades of research, when we look at the maintenance immunosuppressive regimens nowadays, they still rely on the same core medication that was introduced years ago. And we see that the long-term outcomes start to plateau, and patients often experience toxicities of these regimens, and they actually voice the lack of personalization in their care. So to better guide treatment decisions, we actually rely on monitoring strategies like blood tests and imaging. However, these tools often don't reliably detect early signs of graft injury, and it's hard to differentiate between the causes of the dysfunction. And as a result, often transplant centers eventually rely on invasive routine biopsies, which actually carry a risk and sometimes don't yield that much diagnostic value. So actually within today's webinar, we highlight the urgent need to shift to more towards precision medicine and transplantation, and it really outlines the emerging strategies to personalize immunosuppression, but also improve diagnostics and how we can refine monitoring and managing of transplant patients.
So I might ask Alex now, how can we move beyond the one-size-fits-all approach, which Marta's already mentioned, and really implement personalized medicine in transplant?
Yeah, that's thank you, Jennifer. This is actually a key question, just to kind of set the stage on how we started this review. And we are actually trying, and what we tried to do is to address the concepts in solid organ transplant, but kidney, heart, lung, liver, pancreas. We try to be broad in that review. And if we think about the transplant universe we are talking about, let's say that 1.5 million people live around the world with an organ transplant. 200,000 transplants are performed every year, 2/3 being kidneys. And to be honest, I mean, one of the key questions is that the standard of care since I was a resident, which now was many years ago, has not really changed. And Marta was kind of touching on the one size fits all. And the thing that we have learned over the past few years is that there are like numerous technologies, biomarkers, not talking about AI, clinical decision support systems, gene expression assessment for precision diagnostics. All of these tools and this transplant research has been dedicated to address the main unmet needs. And the main unmet needs, I think, are clearly recognized, and we kind of summarize them in the Lancet series paper. Is our gold standard for histology is imperfect, first of all. Point number two, we need to have non-invasive biomarker-guided strategies to optimize patients' monitoring, to tailor immunosuppressive regimen in a manner that will allow the right dose for the right patient at the right time. And we also have to think about beyond the box of rejection treatment to a more pathogenesis-based appraisal of rejection using these tools. And we do have an array, I would say a constellation of things, and it's very difficult to really know what are the key things that will be translated from research to innovation to change patient care and to demonstration that we're improving the patient journey, right? So this is the new approach that we try to frame in the review, and you can see the nice figure which is shown here. And to say that we can advance beyond the standard of care for diagnosis activity, stage, risk stratification, response to treatment. So those are the pillars and the backbone of, I think, what we'll be discussing around this webinar. And I'm really happy, to illustrate this in, this figure that shows actually a transplant patient journey. And we have to think about, and we're going to go deep into the details of biomarkers, but into patient monitoring. We need to reduce the number of unnecessary biopsies. On the other hand, we need to trigger biopsies that will show an increase of the yield, which means the probability that this biopsy will lead to clinically actionable interventions and therapeutics modifications. So all of these multimodal non-invasive monitoring will rely on some biomarker and test, which will be encapsulated in algorithm that will transform this value into patient trajectory, risk, and probability to respond to anti-rejection therapy, or likelihood to benefit from a minimization or a switch from CNI-based regimen to belatacept, for example. And then this is not going to replace biopsies. Biopsies are here to stay. It's just that we need a better use of those biopsies, meaning that it's pretty easy to discriminate from a normal biopsy to an abnormal biopsy. But if we think about rejection and all the concepts that are moving rejection from 2 single entities to now 10 of different archetypes, we will need in some clinical scenarios complementary tools to inform those biopsies which are equivocal for diagnosis, which could be this new MVI concept of injury. And this is where also the new technologies are here with molecular diagnostics in the biopsy. And I think the BANF conference is really trying to go forward and illustrate these important concepts. So it's not to say that we are there yet for wide, broad implementation, but at least there are some ongoing clinical trials following this patient journey, and you can duplicate or copy-paste this strategy from kidney to heart, lung, liver, and pancreas, also with some differences and discrepancies, but a lot of similitude in the global concept. So this is really what this figure illustrates and what we tried to illustrate here, from addressing the value chain of post-transplant patient monitoring, stratification, diagnostics, leading to more personalized treatment. It's becoming pretty obvious now with the new drugs and the pipelines. And let's say that we are talking here about allo, but all these concepts are also applicable to xenotransplant in the field that we are now implementing clinical trials. So we kind of spend a lot of time to frame and design this figure, and I'm very grateful to Marta, Peter, Kiran, and also some people of my team like Fariza. But I think it's really the illustration of the concept that we have in mind to change and to address the unmet needs.
How do you think you said it's not ready yet, but how do you think the next 5-10 years these biomarkers could be used to advance our fields a bit more?
I think now the biomarker, and we can touch base on biomarker, but let's say that biomarker has been a very noisy has been very noisy in our field. So many biomarker study, and we address that in a paper which we released in Journal of American Society of Transplantation of Nephrology. The biomarker landscape in transplantation is just so wide, from blood-based to intragraft to urine-based, combination, other using proteins, cellular assay, RNA, DNA, antibodies, metabolites. And to be honest, I was quite confused. I mean, of course, we have DSA, but DSA does not cover the entire spectrum of injury. So there are a lot of biomarkers. And a lot of the biomarker studies, unfortunately, including my own studies that I did in the past, right? But they did not reach the sufficient level of the methodological standards to prove for definitive adoption for clinical care. And as you know from that GSN paper, most of the biomarker studies tended to be single center, retrospective cohorts. They did not challenge the biomarker of a standard of care, which I call the sick versus well approach that makes all your AUC perfect. And I think that unfortunately, this has created some kind of noise in the transplant biomarker, field. And I think we are now in an era where we kind of understand the pitfalls. And this is also, Jennifer, why we are now, and we kind of touch that also in the review, we are advocating for what I call the DDMT approach: design, data, transparency, methodology, large unselected cohorts, challenging biomarkers of a standard of care, looking into deep dive into different clinical scenarios to see where the biomarker stands and what is the context of use. So in other words, to answer your question, I think we are in a position where we are starting to see better designed biomarker studies. So this is where the context of use can be defined and the real clinical utility or not of the biomarker can be demonstrated. But I guess Marta will provide you some concrete example with liquid biopsy, for example.
And just before we move to Marta, Alex, the last point you brought up was very interesting. Do you think there's a way to encourage sharing within the field between all the researchers without stepping on each other's toes?
Yeah, I mean, Jennifer, that has been the DNA of the team. And, Peter has spent a year here. What we are trying to understand in the lessons is that we need more large collaborative studies. And I think that's the essence of what we have tried to put together. And the paper we published also in the New England on MVI is a perfect illustration. We need data, we need heterogeneity, and we need well-structured and qualified data sets to address the differences in systems, in populations. And I think your point is just very crucial, that this next stage of trials will need some cooperation in data sharing. I don't know if you're aware, but at least in Europe, there is this huge initiative, which is called European Data Space, which is that for the academics, Europe is really trying now to foster big repositories for improving data sharing process. So, we regulate too much in Europe, but at least there is this initiative. So the answer will be towards well-designed studies, and also number matter. And I think the collaborative spirit between centers across different continents is absolutely needed.
As Alex mentioned, when we started this review, we really dove into the world of all the different biomarkers that are available and has been published on. And Kieran has really taken the lead on diving into it and to decide, okay, which biomarkers are we going to highlight within the review, and has been mostly researched. And we know all the traditional biomarkers that are organ-specific blood tests, but also all the imaging that needs to be performed and functional assessment that are made. But I think there are a lot of new molecular biomarkers in the pipeline that are of interest. And I think a key example, of course, is donor-derived cell-free DNA. We've seen that whenever the levels elevate of donor-derived cell-free DNA, there is often an indication that there might be injury going on. And we've highlighted also this biomarker within the review. And I think within the Nature Medicine paper, we also really nicely showed how this biomarker can be of added value within the clinical practice to actually help rule out infection and potential rejection, and potentially reduce the amount of unnecessary biopsies that we might be performing. And we are exposing the patients to. But there are also other biomarkers, of course, of interest, like blood-based gene expression profiling, which has been around for quite a while now, in which you can measure actually the activity of specific immune-related signatures in the blood, and that can help you maybe distinguish between stable and at-risk patients. And when you look within heart transplantation, for instance, this was actually eventually included in the clinical guidelines as a potential alternative for surveillance biopsies in low-risk heart transplant patients. And there are multiple other biomarkers also available, such as microRNAs, chemokines such as CXCL9, CXCL10, which can be measured, for instance, in urine or in bronchial alveolar lavage in lung transplant patients, or microRNA. And a lot of different studies showed promise of some of these biomarkers, but often all of these biomarkers, as Alex has mentioned previously, still need to be validated. We still need to know what actually the added value is of these different biomarkers in the clinic. And I think an important study also within heart transplantation was a study that was published in 2023, which was a multicentric prospective study that actually showed that a pre-identified microRNA signature failed to be reproduced when put into a real-world clinical setting, and it failed actually to reproduce the clinical benefit that was seen previously. And I think it's also very important that we publish these type of studies to really know which biomarkers will help us to provide help within monitoring patients and allow us to early detect injury of the allograft and eventually help to personalize the immunosuppression.
I was struck since I was a resident how the kind of non-invasive people were fighting against the invasive like people, making the histopathologists and the clinicians. They had this dogma that non-invasive biomarker will replace the biopsies. And I think that was completely wrong. And instead of disconnecting, we are now like merging them in a patient journey. That's exactly, Jennifer, what I was trying to illustrate. And we put it that also in the paper, is that in the continuum of the transplant patient care, there is like a room for both and a need for both. We are not opposing them. You will not expect, given the complexity of what can happen in a kidney tissue, where you need to go inside where the disease takes place, that a non-invasive biomarker will tell you everything. So it's this concept has been kind of overstated. And even, biomarker people and also biomarker companies, to be honest, now they realize that biomarker has some context of use, especially for monitoring and many contexts of use, and we can elaborate on that for monitoring, reducing, as I said, number of unnecessary biopsies, deciding which patient could be minimized, and control for anti-rejection therapies, response to treatment. I think there is a room there. But there is a point where you will need that needle. Now that we talk about that, we and the Banff classification recognized years ago that this standard histology is not able to address the iceberg of changes that happens in a kidney. All these biological operational processes are not captured by standard histology. So this is where, in prototypical situations in which the histology or the histopathologist or the clinicians, they are not very comfortable with the diagnosis, you can add these more in-depth tissue analysis with different tools and platforms, and we can talk a little bit about that later. But just to use gene expression in biopsy that will inform not only pathophysiology, but also the disease activity, the main pathways involved, and the main cells which are seen in the graft. And I think that is the key for really moving into a targeted therapies in rejection. And we've learned that from antibody-mediated rejection. Some agents prove to be ineffective, like complement inhibitors, just because we didn't have the right tools to see what was really going on. Meaning that in the same level of MVI, microvascular inflammation, the kidney allograft, the patient under complement blockade had much lower molecular signals. So the histology, and that was not captured by the standard histology. So first point, Jennifer, not disconnecting non-invasive biomarker and invasive. They are part of the continuum. And then if we talk about invasive, not all biopsies will need those fancy tools, right? We have to identify the very clear situation in which we need more. And I think the BANF international classification is working hard on that. There are different platforms, different technologies, older technologies, more recent technologies, some which are more, I think, suitable for clinical implementation, some which are now more dedicated to research, like single cell and paraffin embedded sections. All of these single cell are now gaining momentum. And all these multiplex stainings appear also to be very useful to characterize the cell infiltrates. And that's true also in heart, where we are now better in identifying some of these also rejection patterns. Last not least, I don't know, but if you've probably seen the most recent clinical trials testing drugs like the Felza anti-CD38, you see those technologies used also in those trials. They are using non-invasive biomarkers as complementary tools to see the injury patterns. They are using also gene expression assessment in the biopsy to have a more granularity into the injury process. And I think this is also where the pharma industry is going. So that's good news. These tools are now reimbursed in some countries for non-invasive biomarkers. For gene expression assessment in the biopsy, we're not there yet, but they are also now a big initiative and the BANF classification has endorsed a consensus gene set, which I think, to my point, was a very important step forward to have these BANF human organ transplant panels that encompasses 770 whatever genes, which are now used in many different platforms around the world to tackle the rejection process and move into a more precision diagnostics. And that's true expanding from kidney to heart, now to lung, and liver is a little bit more complicated. So it's pretty good news because I think now we are kind of reconciling these approaches. And for tissue, it's very important. And the last point is about, again, and you know that I'm a little biased about that, but AI is important. With all these images, there is a room for computational pathology, for digital pathology, for automated diagnosis, which is also that we have published also in Nature Medicine, which is the BANF automation system that is the strict application of the BANF rules. So if you think about all these new technologies, I think that many ongoing clinical trials are using this technology and are really showing a great promise. I think these ongoing clinical trials will find room for proof of clinical utility of those approaches.
You bring up an interesting point about clinical trials. So I see quite a few that use it as part of their secondary outcomes or looking for, discovery sets. What about using biomarkers to help select patients? Because one of the things that plagues transplant trials is the number of participants we have.
Yeah, no, of course. I think, all these approaches, I mean, talking also about some surrogate endpoints and predictive systems, I think they can be very useful for patient selection. So first of all, let's say that if we jump into clinical trials, the main issue with clinical trials are physicians, and including myself. We are just a few physicians around the table, and the study is designed based on that, right? And it's eminence-based. And I think now the field is really moving into a more data-driven approach where you have the data from large, well-annotated data set that can help to streamline the inclusion-exclusion criteria, the anticipated patient trajectory. And this is helpful to reduce the sample size, to have a more clear definition of what is the target population that will benefit from the drug. Believe it or not, I'm coming back to the eculizumab trial. 30% of the included patients in the eculizumab trials did not have complement binding antibodies. So again, a simple biomarker that could have said that you needed to include patients with complement binding anti-HL antibodies in a study using as a sole agent to prevent rejection complement inhibitors makes sense, but that was not done because the study was not designed in this way. So this is where I think data-driven strategy is kind of moving forward, not only as surrogate endpoints, and I've been giving many talks about iBox and these kind of things, and not only what we do, but a lot of other teams, they are not developing tools for in other organs, but not as surrogate endpoint, but also as real-world evidence-based comparator arms, synthetic arms, digital twins. I think we are like now incrementally stepping into that. Years ago, the FDA was not aware and of these approaches, and now they are in the process of having now an multimodal algorithm that virtualizes your clinical trials up to 5 years after the randomization, and they will accept as a co-primary endpoint that a predictive algorithm that puts your patient trajectory over the long term for the hard endpoint will be accepted like that. So I think that's a big step. And this is also what we wanted to illustrate, the new paradigm of data-driven strategy for next generation transplant clinical trials. This is also a very important pillar. We are not talking about here about patient monitoring. We are just talking about drug discovery. And to be honest, I think that what I have in mind is that we need to have the industry back in transplantation. We have suffered over the past 10 years that the industry was kind of not investigating and not, having the trials and the momentum in transplantation. And I think that this approach can really trigger the fact that the industry wants to come back to transplantation, invest money on developing drugs. And I think this is all for the patient, the benefit of the patient. So what I'm saying today is, I think, pretty clear, and I sense that the health authorities are now more embracing more and more this concept with a sufficient level of validation, of course. But they have pushed us pretty hard on the qualification process, which made my life kind of a nightmare. But that's fine. But it was very important for us to illustrate that journey from the classical approach and the compromised, BPAR, biopsy-proven acute rejection, which doesn't mean anything anymore, to a more like comprehensive assessment of patients from baseline to the surrogate endpoints.
And Alex, all the work you've done with the iBox for the surrogate endpoint, if we said we find new biomarkers, there's some can be positive, but the effect size is always quite different and context, dependent. What do you really need out of a biomarker? How does it need to perform to be clinically useful?
I mean, you mean in clinical trial, Jennifer, in a clinical trial?
Trial and also just trial and patient monitoring. Trials are a little bit different, but patient monitoring, if I want to add it, what's the value add for this $500 biomarker? What will it add to my patient?
Yes, that's a complex cycle of life for biomarker adoption, and it's well beyond transplantation. And as you know, many steps are necessary. I mean, the key is to prove clinical utility and that you're improving patient well-being and outcomes. And that can be a little bit, difficult to prove, even in some biomarkers which have been reimbursed in the US and adopted, like cell-free DNA. We are now trying to provide more evidence that to understand if this really help patients to live longer, better, with less complications and less immune burden of the immunosuppressive regimens. And of course, you mentioned the price, and we need to prove that it's cost effective for the society, meaning that the biomarker X, Y, Z, whatever, also saves money for our healthcare system in France now. I mean, they are very, picky about that, of course, but everywhere in the world, we cannot afford technologies which cost money if we do not have the proof that it improves not only patient care, but also reduces the costs. So for a concrete example for patient monitoring that we have now, and this study is over, and we are in the process of putting the data together, and we'll be presenting that, by the way, at WTC in a late-breaking abstract, is a randomized controlled trial, really looking at a standard of care, patient management, and biomarker guided strategy with donor-derived cell-free DNA. And we have an economical study. We have primary endpoints on the reduction of unnecessary biopsies, putting the patient in safe conditions, also how we can trigger and how we can augment the yield of those biopsies. So again, I think the good news is that we do have some ongoing randomized trial supporting the use of some biomarkers for patient care, going deep into the level of the proof of clinical utility as well as cost effectiveness. Now for clinical trials, and if I come back to the definition of the biomarker, let's say iBox has different parameters, and you can say that there are some biomarkers here like EGFR, DSA, whatever. So the pure definition of the biomarker, remember, is a characteristic that is objectively measured as an indicator of a normal or pathogenic biological process, or a response to a therapeutic intervention. it's absolutely important, and I've been discussing that over and over, that there is no requirement that if it is an indicator of a normal pathogenic process or pharmacological response to a therapeutic intervention, a biomarker, meaning that the biomarker can have no, meaningful association with a particularly biological process, which open the doors to errors. So GFR is a biomarker, right? But a biomarker of everything, that every single insult that the graft can have. And it's very interesting. So if you consider iBox, for example, you can say that these are some biomarkers and parameters which are combined into a multimodal predictive algorithms. That's why the iBox went into the biomarker qualification plan as a surrogate endpoint. And a surrogate endpoint is how you can use something to predict something, which is a hard endpoint. For example, in iBox is graft loss.
Thank you, Alex. I'm going to encourage the participants, which I can see there are 121 people. That's fantastic. Please post questions into the Q&A. And we have one from Kevin Fowler. Since kidney transplant recipients' data is their own, have you considered activating the patient community to request the data must be shared? Happy for a stakeholder strategy, and he loves your vision, not my vision. It's from Kevin. Has anyone involved the patient community to help drive and push along this data sharing side of
I'm
things?
sure Peter can answer, but if I can just start, and we've been discussing with Kevin for years, and by the way, what Kevin does for the community is absolutely amazing. And Kevin has been also involved in some of our papers and discussions around iBox, for example. So it's very key that we involve patient communities, that we include in our studies patient-reported outcomes, the PROMs, PREMs, all these things. And this is our lesson for ourselves, that we are now very implementing that in all the studies that we are designing. Regarding data, I think, with all the tools and software which are used in a more wide manner, the patients also have data. And I think that we can have some patient engagement and the patient own their data. And the data sharing process that we can gather from patient advocates and this kind of initiative, especially in the US, where there is so much dispersion of the data because the patient do not come back to the center so often, depends on the center, is very key. So, again, very well taken point from Kevin. And we are trying now to address these key questions in all of our studies, involving patients also in the design, having patient advocates part of our committees for designing the trials, and also work with patient associations, that we do that in France and Europe, to have also a comprehensive assessment of what kind of the data we can gather from the patient, including their well-being and all these very important parameters. But I'm sure, Peter, you have specific ideas also about that.
Yeah, I would just say that I think there's been a very important trend inside transplantation and outside transplantation to invite patients to the table right when the study design is being conceived from the beginning. And I think that that's just very helpful. It's extremely valuable to have people like Kevin who really understand the patient experience, but also have taken a deep interest in the scientific questions being asked. I think generally, to Alex and Kevin's point, I think what we're seeing in transplant is that we are benefiting from what I think is a broad global trend towards improvement in thinking about how you evaluate a biomarker with predictive analytics. So I think in the sort of old days of transplant, a biomarker would emerge. It would make sense immunologically in some way, and one center would take a look at that biomarker in their own population, and then people would get excited and interested in it. But I think, with contemporary advances in predictive analytics and biomarker analysis, you see that, first of all, we have to share data because a single center experience will never be sufficient. We're really looking at large populations, multi-center populations, independent validation cohorts, very high quality data, all these points that Alex was making. And so the sort of the interstitial tissue or the glue of that is willingness of centers to collaborate with each other. And so I think that as the field of predictive analytics and the field of biomarker evaluation has, sort of matured and cardiology and oncology, it's exactly the same thing here. And so when we come to the table with patients, multi-center, international populations, then we can actually run a very high quality study and feel confident that we understand how the biomarker works. And it's when we sort of retreat to our single center populations and feel nervous about sharing the data that we kind of get into trouble. So I'm really happy to see this kind of evolution of the field and maturity of the field, and sharing the data is going to be a really key part of that.
To add to that, Peter, I think we're already moving towards that the different centers are willing to share data and see the strength of collaborative efforts. We're working a lot with the US as well, in which we see that these collaborative efforts are really the force of understanding how valuable such a biomarker may be. So it's definitely something we're already seeing in the different publications that have arisen over the recent years.
Yeah, and that requires a lot of efforts. I will say that 90% of my work and problems uh, come from sustaining the network of collaborative centers. It's not easy every day. We need to navigate on the different GDPR regulations. I must say that sometimes it's easier to work with the US than in the center, like 1 kilometer from my lab. Um, Peter knows that very well, but I think the illustration and also Marta is a little bit traumatized because she's working so hard to have this data acquisition and making so many calls. But we're super happy because I think that that's a very powerful, and I see that there are like 120 participants in that TTS forum. We are super happy. You can reach out to us anytime. I think that the collaborative spirit is the new way, and how data science is aggregating, I think, more collaborative because we are just stronger together. So it's an appeal also that I would like to make towards this collaborative effort. And I think now the data sets also can benefit from many other questions that anyone can have. And that's also what we are trying to push in terms of some ideas. Not to say that anyone has access, but at least people working in the global consortium, they can use this data set as validation purposes for their own trials. So I think, Jennifer, it's very important to notice that this is the new, I mean, data is oil, it's soil, it's like everything nowadays. And also different data generated from different platforms as well with cutting edge technologies. When you merge all of that, I think that it's very key. Last point that I wanted to make, which is a very kind of hot off the press. Maybe you've seen in the US that the NIH issued a text and released a note that said that they will not accept grants working only on animal models. It's a complete shift in paradigm where to say that if you do not have human data, you will not be able to do some research at some point. So we need to also to cope with the evolution of our societies and the evolution of the politics. And it stresses the need for the integration of different approaches and not to opposing them. I was, having great discussions at ESOT with Fadilakis that you probably all know, that says that, oh, data science is kind of bullshit. It's just pressing one button and we find everything is significant. And I did a PhD in cell biology and this one in statistics, and I could see these oppositions also from the statistical people, the statistician that say, oh, basic science, they are comparing one mice, two rats, and they have P of 0.00949, and they just wait for an additional day for their box to see the cells surviving or dying. I think the truth is in the middle, and I think talking about data, we need to understand that we navigate in a world where more and more health authorities will be seeking for human data, well-informed cohorts. So that's a very key point, and I hope that we address that in a sufficiently clear manner in the Lancet series, but we tried at least.
I forgot to introduce in the introduction to say Alex has 2 PhDs, so he's Dr. Professor Alex Loupy. And not only he did it in epidemiology, he did the hardcore way, which is science, and brought back from PTSD for me, who I just finished it not long ago. I concur with everything you guys have said, And I say that in Australia, to try and share data has immense amount of red tape, which is, I don't know why, but it feels worse than international standards. So I wish the policymakers around the world would make it easier to collaborate. We'll move back to some questions. So Gilbert Burkhardt has said, what could kickstart the involvement of industry and FDA in this biomarker qualification process? An FDA workshop? The last one in transplant was 10 years ago.
Yeah,
I'm
I
sure.
mean, a very important point. I was actually in that FDA meeting 10 years ago, and here I am, now with iBox reaching the stage of full qualification. We are not there yet, but we are making progress for the co-primary endpoint. So I think that the AI field is now, I mean, where we are nowadays is not what we were 10 years ago. There was a lot of suspicion of, models and predictive algorithms. And I think that now the FDA will be well aware that you can have very clear, data with transparency and not the black box effect. And they will be willing more and more to adopt those strategies. So to kickstart the involvement of the industry, I think that this qualification will be a very fresh news for the transplant community. And I think that now the industry is like looking closely, and to kickstart, let's wait for an additional few months and see how this potential qualification of iBox will leverage the interest of the industry back into the process. And yes, an FDA workshop will be great. And Gilbert, last year, I was very pleased to notice that there was an iBox dedicated FDA meeting at the American Transplant Congress. I was not giving any talk there. It was other people talking about the journey. And I saw that was a very positive sign. And I guess that if you look at the WTC program, you might find some session like that. So I guess that the FDA is now willing, to embark upon those approaches.
And RM Han asked a question which follows on really. With all these different biomarkers and technologies, integration of multiomics and AI, etc., how will the transplant physicians and surgeons be trained to keep pace and make use of this really and understand it?
Yeah, well, I mean, Marta is a very young MD and a PhD candidate. So I had this discussion with my students actually. I think this change in paradigm is also changing the way that we'll have to teach, physicians how to cope and how to cooperate with these AI tools. I think that's key. And unfortunately, I think the universities, most of them, they are not like very well aware that their program is kind of embarking upon those new approaches. But I think this is very key that we make people understand, learn, and how to cooperate as companion tools with this multi-omics approach that I digested by some AI and give some probability in a dashboard. We need some efforts. We need to train also with the level of understanding, just to be able to override also some recommendation that AI can make, which can be a trigger for actually bad decisions. And we've seen, example with EPIC proprietary sepsis, prediction system, which created more harm than benefits. So I think that we also have to be very careful in a way that the way we train people, we need to also teach them the layer of methodology that will allow them to have a critical view on those tools and not to take them as granted. But there is a major paradigm shift. New creatures of physician will be created, and I call digital clinical scientist. It's the way that you are a fully trained physician, but on the same way, you know how to use these tools and to also challenge them.
Yeah, and to add to that, I think at the latest ESOT Congress, Professor Segev also really nicely demonstrated that, saying, for instance, AI should not be seen as something that will take over our job, but should be seen as a partner. So I think the future clinicians should actually see it as a partner and how to actually become better at their job and how they can use AI to improve patient transplant care in this case, instead of seeing it as a tool that will take over and critically actually understand and analyze the information that these type of tools may provide you. So I think it also needs to be a paradigm shift towards being a partner with these new tools rather than seeing them as potentially in opposition. But of course, education is part of that, and we need to teach people how to understand these different tools and what does it mean to have a different score regarding prediction, to have rejection, and how do I interpret that as a clinician. And I think that's definitely something that needs additional attention.
I think it's also, when I think about the ability of these augmented intelligence tools to integrate large amounts of data, I think it just provides another reason for optimism. I think one sort of scenario that you can imagine is you're rounding on the inpatient consult service with the best fellow you ever had. Because, if someone sits down with the chart for an hour and looks at the trajectory of all the biomarkers that are currently available to us and comes to you with a really thoughtful story, having integrated all that information, you as the attending will make a better decision. You'll be better prepared because someone has done all that background homework for you. Now imagine that you're in clinic and there's, an augmented intelligence dashboard waiting for you, where there's not going to be anything that's ever going to replace your ability to make a connection and talk to a patient who's going to tell you how they're feeling and tell you things that are not in the chart. That's what you will bring. But what I think the dashboard can bring is some integration of new bio and old ones. So there's the patterns and the intensity of the donor-specific antibody. Maybe now there's the cell-free DNA. Maybe what will emerge is new patterns of interpreting low titers of viruses that we commonly think of as, an infection, but might instead be thought of as a measure of immunosuppressive adequacy, like CMV or something like that. So bringing all that together with patient after patient, I think we'll come to those encounters more prepared. But I think to the question that was asked, I think it is going to be an ongoing challenge for all of us to remain open-minded to these new tools, to accept them and to sort of, kind of continually appreciate their strengths and their weaknesses. But certainly continuing education is going to remain a very fundamental part of our practice because the new knowledge will arrive very quickly. So thank you, Dr. Hahn, for that question.
100%. And also, Menak Ashkar has also asked a question and follows on from the previous discussion about cost effectiveness in the consideration of biomarkers, and how if anyone can comment about consideration of LMICs and how they can be engaged with this to move this forward globally.
Yeah, I mean, if I can, hi Medat, nice to see that you're connected. Looking forward to see you at WTC and Ashi. So for low and middle income countries, I think there is this paradigm where we think that AI will be like too costly. We have concrete example of deployment of LLM in Africa to target specific population for screening for hypertension or renal diseases. So it's not like biomarkers, but it's sort of like a more general consideration that some of those AI tools and those predictive tools can be perfectly well suited actually for low and middle income countries. Look at the virtual biopsy system that actually you just need simple donor parameters, and with machine learning, you can predict what the biopsy will look like and the BANF lesions. This is perfect for low and middle income countries. They cannot afford to do so many like pre-implantation biopsy. So they just to use an AI tool, which is openly available, and they have their biopsy without the need to do the biopsy. So I'm just trying to replace that in the fact that AI and innovation is always perceived as something very expensive and not adapted to low and middle income countries. I think in the contrary, that some of the AI tools, predictive tools, will be largely supported by low and middle income countries for their broad adoption and use. Everyone in Africa, they have smartphones. They were starting to see that, to think that no, in Africa, that's not going to work. Oh, guess what? I mean, they all have smartphones right now. So those LLM-based campaign, and they deployed agentic AI for screening populations, works perfectly well there.
Yeah, I think the question of equity is a very important one. We're going to take that on with our next webinar and try to answer this question as well as we can. I think in general, we should be optimistic that a rising tide raises all ships, so to speak. So I think the the quality of care over the next 10 years for all transplant recipients should get better. I think the expansion of the organ supply should continue. I think there's a separate question. Everyone's situation might get better, but whether or not the situation in the global north and the global south widens is kind of a separate question that we will definitely tackle. And I think it is very important whenever we can to ask ourselves, how can the benefits of a new technology really accrue to the people who need it the most?
Great. So the last two questions relate towards biomarkers to rejection diagnoses. Mohammed Habel had a question about, is there one that's predictable and specific, highly specific and sensitive to, rejection close to tissue biopsy? And Yassar Kaliskan asked something similar, but is there some way to differentiate ABMR and TCMR with biomarkers?
Yeah, I mean, what you are really expecting from monitoring biomarker is that it captures rejection process. And I think, what we've demonstrated with donor-derived cell-free DNA is that it's very performant for predicting biopsies that shows either AMR or T-cell mediated rejection. Point number two, I think that you will never replace a biopsy because even inside T-cell mediated rejection, you have different archetypes that will not need the same, care and treatment. Last but not least, you're never gonna treat a biomarker. You're gonna treat a biomarker that triggers a biopsy that shows a disease. So we must be very cautious on that. But again, I think we do have some pretty good biomarkers that have shown their very important association with the TCMR process, but not giving you the granularity of information that you will need to just decide what is the burden of increase that you need to do with steroid pulse or thymoglobulin or increase CNI or just switch to other drugs. Again, some biomarkers very well correlated in acute cellular rejection, but they will not be replacing the need for the biopsy. Last but not least, all those biomarkers were trained on the imperfect gold standard. So a bunch of the biopsy, which are labeled ACR, might be more complicated. So it's a kind of schizophrenic way of thinking, is that you train biomarkers to predict something, which is the imperfect gold standard and maybe some of the mislabel. And now the more we understand a biomarker, the more we are using the new tools to understand that some of the mismatches, let's say that false negative of biomarkers, which means that they are negative and the biopsy shows like some features of inflammation, molecularly look like AKI and not rejection. So it's the imperfect gold standard process also that we need to be a little bit cautious about.
Fantastic. Thank you, everybody. So this comes to the end of the webinar, and thank you for all our participants, to Alex, to Marta, and Peter. Just to plug for our next webinar happening on the 25th of July in a week's time. It'll be with Dr. Elmi Muller, Dr. Lisa McElroy, and also Peter again, to discuss the policy innovations to advance equity in solid organ transplantation. So I encourage you to register for that and also to answer the survey which Roberto has published in the chat box for your certificate. Thank you, everybody.